Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjon classes. Dear student, we are in a continuation of the chapter that is Anatomy of Flowering Plant, that is Angiosperms. And in that, in a previous class, we have already covered with the meristematic tissue in detail and the type of meristematic tissue or meristems. Today, we are going to talk about the permanent tissue in detail. Okay. So first, by hearing the term permanent means are those cells which are fixed, means which have stopped their capacity of division and they are now in a non-dividing state, then they are called as a permanent tissue. Okay, so it is composed of cells which have lost the power of division temporarily or permanently, and this permanent tissue are actually formed by a division, or we can say a differentiation of the meristematic tissue, and the cells of permanent tissue do not divide generally further. Okay, so these cells of the permanent tissue do not divide further as we have discussed, but this permanent tissue are of two types according to NCRT, which we are going to study, and this permanent tissue which have lost their capacity of division permanently or temporarily are divided into two groups, and that is simple permanent tissue and the complex permanent tissue. Now this simple permanent tissue, if we talk, this permanent tissue have all the cells similar in structure and function, means simple permanent tissue are made up of, we can say, a one type of cell, or we can say they are made up of structurally similar type of cells that perform a common function. But complex permanent tissue, if we talk, they are, or this permanent tissue have many different type of cells, means they are made up of a different type of cells. So simple permanent tissue are made up of one type of cells. Because of this, they are also known as homogeneous tissue. They are also known as homogeneous tissue. And complex tissue are made up of different type of cells. So they are known as heterogeneous tissue. Okay. Now, if we discuss the simple permanent tissue and complex permanent tissue further in detail, then first we are going to discuss simple permanent tissue. This simple permanent tissue are made up of only one type of cells, which are structurally single type of cells and perform a common function. And this simple permanent tissue are further divided into three types, that is parenchyma, collenchyma, and sclerenchyma, which we are going to discuss here. When we discuss first the parenchyma, this parenchyma are the cells which are a major component of the organ. Means if we talk about the plant body and if we take any organ of a plant, the major component or the major tissue which is found in that particular plant organ or a plant body, that is nothing but a parenchymatous tissue. And this parenchymatous tissue, if we talk about their cells are generally isodiametric, means the diameter of each cell is almost or approximately similar. Then the parenchymatous cells, if we talk, their shape, their shape may be spherical, means they have cells may have a spherical shape. They can have a oval, round, polygonal, or elongated. This many shape can be found in case of this parenchymatous tissue, or the cells of the parenchymatous tissue. You can see this is the parenchymatous tissue, and in this you will see the presence of intercellular spaces between the cells. You will see the presence of intercellular spaces. So if we talk about this particular parenchymatous cells of this, then we can say that parenchymatous cells may either be closely packed or have intercellular spaces. May either be closely packed means they do not have intercellular space, or they have small intercellular spaces means intercellular spaces may or may not be present. They may or may not be present. Further, if we talk, they have thin cell wall and a cellulosic cell wall. Cell wall in parenchymal tissue is made up of cellulosic cell wall. They may be either closely packed or have small intercellular spaces, just we have discussed, we have written. The parenchyma perform various functions like photosynthesis, storage, and secretion. According to NCERT, these three functions are the important. They perform photosynthesis, storage, and secretion. These are the intercellular spaces which may or may not be present. Okay. Now, apart from this, if we talk about the parenchyma, then parenchyma are very primitive type of tissue. Very primitive type of tissue, or we can say the first evolved tissue. And it is known that remaining different type of tissue are derived from this first evolved tissue, that is parenchyma. So we can also call parenchyma as a fundamental or precursor of other tissue. Precursor of other tissue, and it is a major component of other organ. This parenchyma term, if we talk about that term coined by Grew, the name of scientist is Grew who discovered this. Now, if we talk about the further characteristics of this particular parenchyma, if we talk about characters or characteristic feature of this parenchyma in more detail, then we come to know that this is a living tissue, and this living tissue is the first differentiated tissue from the meristematic tissue. Then it is also a universal tissue, and the cell is cell wall, thin walled, cellulosic cell wall, mainly cellulose. They may have a pectin. This parenchyma is, we can say, soft tissue. They have large, each cell have large central vacuole, and due to the presence of large central vacuole, the main function of parenchymatous cell is storage of food. Okay, the main function, if we talk about the main function is storage of food. Okay, and we have studied that there is a major component within organ, means it is found in cortex, pericycle, medullary rays, peak, leaf, mesophyll, everywhere you will see the presence of this. Shape behavior we have discussed, isodiametric nature we have discussed. Now we are going to talk about the modification of this parenchyma. Modification of parenchyma. And if we talk about modification of parenchyma, then in this, the first type of modified parenchyma are known as prosenchyma. If we talk about the prosenchyma, the cells of this parenchyma are thick walled. Prosenchyma have a thick walled, long pointed, long with pointed ends. Okay, and this parenchyma, this modified parenchyma form pericycle of fruit. And if we talk about their thick walled, as we have discussed, if we talk about the function is obviously to provide a strength. Apart from this, the second type of modified parenchyma is erenchyma. Now, if you talk about erenchyma, this is made up of made up of rounded cells, and due to this, they have large air cavities. They have large sized air cavities or air chambers. And this erenchyma, if we talk about, they are found mainly in the cortex region. But if we talk about function, then they provide buoyancy to hydrophytes. Buoyancy is a capacity to float to hydrophytes or aquatic plant. After this, the third type which we can see the in a modified parenchyma is chlorenchyma. Now, if we talk about this chlorenchyma, they contain abundant quantity or large number of chloroplasts. They contain this parenchyma contain large number of chloroplast in their large number of chloroplast in their cells, and they are found in mesophyll cells of leaves. They are found in mesophyll cells of leaves. So the function of this chlorenchyma is to perform photosynthesis. Apart from this, if you talk about another type, that is a fourth type, then we call it as a what? Stellate parenchyma. Actually, the first three are too much important. The stellate parenchyma usually you will not find in exams. Okay, and this are a star-shaped parenchyma. We can say what they are: star-shaped parenchyma. And this parenchyma found in leaf bases of banana. They are found in leaf bases of banana, and as we know that we have discussed, the leaf base of banana is sheathing leaf base. A sheath like leaf base is found where the leaf base cover the stem wholly or partially, which we have discussed in the morphology part. So that sheathing leaf base is found in banana, and this in sheathing leaf base you will see the presence of the stellate parenchyma. So means if we talk about the function of stellate parenchyma is to provide a mechanical support. Okay. And the fifth type is mucilage parenchyma. Mucilage parenchyma, and this mucilage parenchyma have a large vacuole and mucilage are present. So they have large vacuole and they store mucilage. Mucilage is a one type of sugary secretion. And this mucilage parenchyma is found in succulent xerophytic plant. Found and in succulent xerophytic plant or a succulent xerophytic plant like aloe vera, opuntia, cactus, and that help or function to store water. Okay, so these are the five different types of parenchyma, modified parenchyma, which we have discussed. Now, at last, after discussing the different characteristic features and the modification of parenchyma, we are going to discuss the function of parenchyma. Dear student, initially in anatomy term we have just discussed that anatomy does not deals with only the structural part, it is also going to deal with the function part. So in each tissue we are going to discuss the function as well. So the function of this parenchyma, if we talk, then the main function as we have discussed, it is storage of food, and some cells also perform other functions are like photosynthesis, secretion. These are the function, but main function is storage of food. So these various functions are performed by the parenchyma, but some cells store waste material. Some cells store waste material, and these cells are called as idioblast. These cells are called as idioblast cells, and this idioblast cells store waste material like we can say it store oils tannin crystal, oil tannins and crystals. And the example we particularly, if you talk about these functions are, these are the all almost important functions we have discussed. We remember in that also photosynthesis is performed by the chlorenchyma as we have discussed. So this is all about the parenchyma which we have discussed detail. Now we are going to talk about the collenchyma in detail, another type of simple permanent tissue which is made up of only one type of cell. Now the collenchyma occur in layers below the epidermis in dicot plant. In dicot plant, you will see the presence of collenchyma below the epidermis in the layer called hypodermis in mainly dicot stem. So we will study that what is hypodermis. So layers below the epidermis in dicot plant means in hypodermis of dicot stem. In monocot stem hypodermis, there is a presence of sclerenchyma that we will discuss. Now, if we talk about the parenchyma's characteristic feature, you will see that they consist of a cells which are much thickened at the corners. This parenchyma made up of cells which have a thickening at the corner, and this thickening is due to the deposition of some material like cellulose, hemicellulose, and pectin. This cellulose, hemicellulose, and pectin are deposited at the corners. So, thickening at the corner you can see in case of this particular collenchyme. Okay, you can see here they have a large vacuole. Okay, they have a nucleus, peripheral nucleus, because large vacuole is developed, and the angular wall thickening you will see present here. It is not clear, but you can see like this. Okay, now the collenchymatous cells may be oval, spherical, or polar. The different various shape can be there, and they often contain chloroplast, so they have a particular function of photosynthesis. So they assimilate food when they contain chloroplast. Intercellular spaces are absent because they are mostly in a polygonal shape and they are closely packed. And due to the thickening of wall at the corner, they provide a mechanical support to the growing parts of the plant, such as young stem and petiole of a leaf. Okay, you will see such particular conditions. Now, apart from this, if we see the particular thickening, they are providing a mechanical support to the petiole. The flexibility to the petiole is also provided by the this particular collenchyma because it is an elastic tissue. And you will see the presence here. You can see the particular corner thickening. You can see here. This thickening are found here. Okay, this thickening are due to the what? Particularly, we can say the collenchyma. Okay, they have thickened corners and intercellular spaces are absent. So if we analyze this all characteristic here now, then this collenchyma term, if we talk, this term coined by Sladen. This term coined by Sladen. And when this term is coined by a Sladen, if we talk about the main characteristic of this particular collenchyma, then we come to know that this is a living elastic mechanical tissue. As we have discussed, it is the cell wall is not completely thickened. It is thickened due to the cellulose and hemicellulose deposition, but at the corners only. Angular thickening is observed. So they provide mechanical support as well as some elasticity. And as we have discussed, they are made up of more or less elongated cells. In a transverse section, they can appear as a oval, spherical, or polygonal in shape. They have angular deposition of the pectin, cellulose, and hemicellulose. Mainly pectin deposition occur. As we have discussed, the intercellular spaces are absent in them, and as we have discussed, they have contain more chloroplast and they assimilate food as well. That all point we have discussed, but they are living as well as elastic mechanical tissue, which we have inferred or concluded after discussing these points. Now, if we talk about the occurrence of collenchyma, that part is important because from this you will get a question. If we talk about occurrence of collenchyma, collenchyma is not a universal tissue. So it is found in stems of herbaceous dicotyledons, herbaceous dicot, that is, we can say young dicot stem below epidermis, as we have seen below epidermis, that is in hypodermis, as a homogeneous tissue, as a homogeneous layer, that is in sunflower stem, or in patches, as in cucurbita stem. Such a way it is seen. So this cholenchyma occur in a below epidermis, means in a hypodermis region of the dicot stem. Now cells of cholenchyma are flexible, obvious, due to the hydrophilic nature of pectocellulose, which can be soluble and which provide us flexibility. Apart from this, cholenchyma is also present at margin of leaf lamina and petiole of leaves. Okay, and when it is present at the margin of leaf, it protects from the action of wind or a cracking which is produced by the wind, that can be prevented by the presence of this cholenchyma. One important point with respect to cholenchyma is that it is absent in mature woody plant parts. Important function that you should remember: absent in mature or woody plant parts. In roots, woody plant parts, roots, and monocot. So it is absent in monocot as well as root and a woody plant part. Now, if we talk about the function, the function is cholenchyma perform both functions. It perform a particularly function of providing strength, that is mechanical support, as well as, if we see, it provided tensile strength. It provided tensile strength against bending. It also provide mechanical support to growing parts of the plant, such as young stem, and petiole of leaf. Due to presence of chloroplast, they can perform photosynthesis as well, which is the vital function. So two functions are there. One is mechanical support, another vital or biological function is the photosynthesis. Okay, so this way the cholenchyma is important. Now, after cholenchyma, we are going to discuss another type of tissue, simple permanent tissue, that is a sclerenchyma. When we talk about the third tissue, that is sclerenchyma consists of long, narrow cells with thick and lignified cell wall. Now, the question is why they have a thick or lignified cell wall? Because the sclerenchyma are the dead or non-living tissue. And this dead or non-living tissue are the sclerenchyma, And this sclerenchyma term is given by Metenius, or term coined by Metenius. Okay, so they have a thick and lignified cell wall, and due to the thick lignification, the cells are going to be dead. Apart from this, if we discuss, they have a few or numerous pits. They have a numerous pits or pits are present. Now, pits are what? Unthickened areas of the secondary cell wall. So pits are the unthickened areas of the secondary cell wall. They are present. And in this, the protoplasm losses occur. So usually they are dead and without protoplast. So you can see this is the thick cell wall they have, and this is the lumen which is almost reduced in case of fiber, sclerenchymatous fiber, and this is the sclerenchymatous sclerids which have a thick wall and they have a reduced, almost reduced lumen. And in this, you will see the presence of branched pits. And in NCRT, a diagram is given where branched pits are or obtained, and these are pits are long or elongated. So the question is, what are pits? That is the important. So we can say pits are unthickened areas of secondary cell wall. Actually, secondary cell wall is laid inner to the primary cell wall. So at some areas you will see the presence of primary cell wall and secondary cell wall both. But at some areas you will see the presence of primary cell wall, but secondary cell wall is absent. This region are known as pits. Okay, and pits are elongated in the sclerids, and they are present numerously in case of this sclerenchyma. Now these jute fibers, okay, and ham and flax, all you, if you talk mainly these jute fibers that are also one type of sclerenchymatous cells, sclerenchymatous fibers. We'll discuss that. The basis of variation in form, structure, origin, and development, if we talk, the sclerenchyma are of two types on the basis of this form, structure, origin, and development. They are of two types: sclerenchymatous fiber and sclerids. If we talk about the fibers, see, these are the dead tissues. They are mechanical tissue, so they are going to provide a what? Mechanical support, and they can be fiber or they can be sclerid. If we talk about the fiber, this fiber are obviously thick walled, but they are fiber. They are named as fiber because they are elongated and have pointed cells they have. And one more important point, that these fibers generally occur in group in various parts of the plant. They are found to be occur in a group. Then the sclerids, if we talk, they are spherical overall cylindrical, and they have obviously highly thickened cell wall, but they have very narrow cavities, narrow lumen, obliterated lumen, almost reduced lumen. You will be seen in case of this. Okay, so these are the division points. So if we talk about this, these are commonly found in the fruit walls of a nut, pulp of fruit like guava, pear, sapota. You will see the presence of this sclerids. Okay, in some legumes, seed coat of legume and leaves of tea, they are also particularly have this sclerids. Okay, these all are the particularly sclerids. Sclerenchyma provides mechanical support to organ. Okay, and these are the particular different type of sclerenchyma. Now, as we have discussed about this sclerenchyma, they have long, narrow, thick-walled, lignified cell walls without protoplast. They are dead. Okay, and they have two types, that is a fiber and sclerenchymatous fiber and sclerids, and the particularly different bases we have also discussed. In sclerids, we have discussed they have small, dead, extremely thick wall, highly thickened cell wall hair. They have they occur in various shape. They are found in the pear, sapota, and in their pulp, also in the seed coat of some legume, as we discussed in case of the sclerenchymatous fiber, which are thick wall, pointed, and in that also lumen is reduced. So this is all particularly different things with respect to the particular sclerenchyma. But in sclerenchyma, as we discussed, they provide mechanical support. The occurrence, we have to note it down. So with respect to occurrence, if we talk, they are found in hypodermis of monocot stem. And we will discuss in later part that where the hypodermis is present, actually it is present below the epidermis, and it is found in mainly in stem. In root, you will not see the presence of this hypodermis. Okay, so this is the thing which is observed. Okay, so this is all about the simple permanent tissue. Now after this, we are going to talk about the complex permanent tissue. Before that, let's discuss some of the question. In which type of cells do not divide further? Obviously, we have discussed that permanent tissue in these cells, the cells do not divide further because they have lost their capacity of division. Same way, the second question is the tissue with many different type of cell is, as we have discussed, the tissue with many different type of cells, that is nothing but a complex tissue. The tissue which show division of labor is nothing but a parenchyma, because in parenchyma different function it can carry out. Cell wall of sclerenchyma are made up of, in sclerenchyma there is a lignin deposition, thick lignin deposition, due to which they become a dead cells. When we talk about collenchyma, it is present in, actually it is present in the dicot mainly, and that is in the stem, in root of dicot and monocot. In root it is not found, as well as in monocot it is not going to be present. Okay, so till now we have discussed all important information, all the points, all the question which can be asked, which type of question can be asked, that also we have discussed in the simple permanent tissue. Now we are going to discuss the complex permanent tissue in detail. So let's talk about the complex permanent tissue. Now, when we talk about complex permanent tissue, these are the tissues which are made up of a different type of cells. They are made up of different type of cells. So these are the another type of permanent tissue which are made up of more than one type of cell, and or we can say different type of cell, but this work together as a unit. They work together as a unit. And in this complex permanent tissue, two tissue we can study in detail, that is a xylem and phloem, which are types of this complex permanent tissue. So complex permanent tissue include a xylem and phloem, and we'll be going to discuss this in detail. Now, before we discuss xylem and phloem in detail, we remember this complex permanent tissue is also known as heterogeneous tissue, and this complex tissue or xylem and phloem or vascular tissue are found absent in gametophyte. You know that. And in plant kingdom, we have discussed that there is a two types of bodies, that is gametophyte and a sporophyte. So this gametophyte found absent in, we can say, this xylem phloem vascular tissue are found absent in the gametophytic body. Now, why it is happening like this? Because the vascular tissue evolved from the pteridophyte, and from pteridophyte the sporophyte is dominant. So these are found in a particularly mainly a sporophytic body of pteridophyte, gymnosperm and angiosperm, not in gametophytic body. So it is found absent in gametophyte. They are absent in gametophyte. And it is known that during the vascular tissue formation or vascularization in plant differentiation, the formation of primary phloem and primary xylem occur in the initial part, but later on the secondary tissue, secondary xylem, secondary phloem are formed during the secondary growth. And this xylem and phloem are the conducting tissue, vascular tissue, which carry out a transport of water, mineral, and the food. So if we discuss in detail about the xylem and phloem, xylem function as a conducting tissue of water and minerals from root to the stem and to the leaves. Means it is carrying out a mainly a particular part of transport of water and mineral. Now this, if you can see in this particular diagram that in veins this xylem and phloem are found. So in veins this water is going and transporting like this. So if we talk about this particularly, that this is the particularly xylem, and this xylem help in a particular conduction of what of water and mineral. Apart from this, xylem have some dead cells as well. Xylem is a tissue, so it have a different type of cells. They have a living as well as the dead cells. So if dead cells are there, then they also provide a mechanical strength to the plant parts. They also play a role in providing mechanical support. Now we remember that this xylem is there. Now this xylem term was coined by Nageli. This term coined by Nageli, and its conducting parts or elements are called as headrome. So headrome is a term used for the conducting parts of the xylem, and we will discuss that trachea and vessels are the conducting parts of this. Now xylem is composed of four different type of elements or four different type of cells, that is the tracheids, vessels, xylem fibers, and the xylem parenchyma. Now when we talk about this tracheids, okay, and the vessels, which are the two main conducting elements, these vessels are, if we talk, they are also known as trachea. They are also known as tracheae. So we remember there is a difference in the tracheae and tracheids. Okay, trachea is the vessels and tracheids are the first conducting element. Now, if we talk about all this, out of this tracheids, vessels, xylem, fiber, if we talk, then this tracheids are dead cell or non-living. These vessels are also dead or non-living. This fiber are also dead or non-living cells. But xylem parenchyma is the only living component of this xylem or element or cells. Okay. Now we are going to discuss this one by one. Dear students, out of this four, in gymnosperm you will see the absence of other three, that is tracheids, fibers, and this parenchyma. But vessels are going to be absent in this xylem of gymnosperm. But when we talk about angiosperm, in angiosperm all four are found. And in these, the first tracheids, if we talk, they are elongated tube-like cells. And this elongated tube-like cells have a thick and lignified cell wall. Now, due to the thickening and lignification of cell wall, the protoplast loss occur, and due to this, the protoplast is going to be absent. They also have a tapering ends. They also have a tapering ends. So these are dead and are without protoplasm, because protoplasm loss occur and they become dead at maturity. So these are the four component. Out of this, the first one with the tapering ends are the tracheids. You can see like this. Now, if we talk about the inner layer of the cell walls have a thickening which vary in form, means they have, in flowering plants, tracheids and vessels are the main water conducting elements obviously, and they have pits. Okay, so you can see here that this is the trachea. and this trachea having this particular space and through which water is conducting. But these are the vessels. Okay. So how they are differentiating from each other, we will discuss here now. So first, we are going to discuss the trachea in detail, which are the trachea and vessels, which are the conducting elements. If we talk about first tracheids in detail, then dear students, these are the primitive conducting elements of xylem. They are the considered as a primitive conducting elements of xylem. And this tracheids are considered as a unicellular. They are made up of single cell. And as we discussed, they are elongated tube-like cells with a thick and lignified cell wall, and they have ends are tapering, and they have a narrow lumen due to this large lignification and thickening of cell wall. Apart from this, tracheids are unicellular, and it is known that they are arranged or found one above the other. They are found one above the other and separated by a cross wall. Separated by cross wall. And this cross wall and cross wall separate them, and this has which has bordered pits means this pits are when pits are a secondary unthickened areas of secondary cell wall as we have discussed, but when these pits are flask shaped, so this flask shaped pits are known as bordered pits. So this flask shaped pits or bordered pits are found in the end walls of the tracheids. Now in tracheids, one more important point is they have lignified cell wall obviously, but they are dead element because they are without protoplasm, because protoplasm loss occur at maturity, and actually the pits are present, so pit are not porous. Okay, so actually you please, if this is a trachea, suppose, don't consider the trachea as there here there is a pores. These are not pores, these are the pits. And as I have told you, pits have a primary cell wall, but secondary cell wall is actually absent. So tracheids, if we talk, tracheids are imperforated, and imperforated means they are not porous. Okay, now these tracheids, as we have discussed, obviously they are unicellular and they have different type of pits. Now the deposition of thickening, or we can say a lignin deposition, vary in form. And when lignin deposition vary in form, it occur in a different ways. So this deposition of lignin on a cell wall is responsible for the different formation of different type of thickening. So the lignin deposition vary in form and it is of different type. So if you talk about, this is of actually a phi type, vary in form, and it is of phi type. So now we are going to discuss the phi type of lignin deposition. So So different type of lignin deposition are found. In this, the first type is annular. And in annular also you will see that the thickening is here. Suppose this is a trachea. And in inner wall you will see the thickening at internal intervals. Then this thickening at certain interval is known as annular type of lignin thickening. And second is the spiral. And in spiral also, and in that also, if you can see this spiral type of thickening, then it is known as spiral. The third type of lignin deposition is scalariform. The scalariform is ladder-like. And in this ladder-like thickening, you will see that the thickening is somewhat like this. So when this ladder-like thickening occurs, then it is known as what? A scalariform. Then the fourth type is the reticulate, and this reticulate is a what? In this a Neet-like deposition of lignin occur. And when this Neet-like deposition of lignin occur, you will see that in trachea the whole Neet-like structure is going to form. Okay, so this is the reticulate type. And the fifth type is pitted. And in this, you will see the presence of pits at the end walls. You will see the presence of pits, that is the unthickened areas of secondary cell wall at the pits. Then it is known as pitted type. Now it is seen that this annular and spiral type of thickening are found in protoxylem, means first formed primary xylem. Okay, whereas scalariform is a ladder like, and reticulate and pitted, if you talk, these are found in metaxylem. So annular and spiral thickening are found in protoxylem, whereas reticulated pitted, mainly pitted type, is found in metaxylem, whereas scalariform ladder like thickening is that it is found in metaxylem of pteridophyte. It is found in metaxylem of pteridophyte. And cycus, that is a gymnosperm. Out of this, the maximum deposition of lignin is found in a pitted type of thickening, and this pitted type is considered as the most advanced type of thickening. Because of this, mainly we see the presence of pits in the different components. Okay, and this tracheids, if we talk about this occurrence, they are found in all pteridophyte If we talk about tracheids occurrence, then they are found in xylem of pteridophyte, gymnosperm, and angiosperm. Okay, so this is all about the tracheid, a dead element. Now after this we are going to talk about the vessels. And when we talk about vessels, vessels which are also known as tracheae, as we have discussed, these are also a long cylindrical tube-like structure made up of many cells. Means it is a multicellular structure, whereas tracheid was a unicellular. And here each with a lignified wall and large central cavity. Here also protoplasm loss occur. Here also these are the dead cells. So the vessel cells are devoid of protoplasm, so they are dead cell, and the vessels member are interconnected through the perforation in their common walls. Okay, you will see the perforation in their common walls, and this is the one way only particular water and minerals are getting transported. Okay, so this is the particularly, and they have thick wall. Okay, so this is the some of the characteristic of vessels. Now, after this common functions, common observations or functions, if we discuss these vessels or xylem vessels in detail, then these vessels are the advanced conducting element, advanced conducting element of xylem. And these vessels are the long cylindrical tube-like structure. Okay, these vessels are the long cylindrical tube-like structure, and as we have discussed, they have a lignified cell wall, but due in spite of presence of thick lignified cell wall, they have wide lumen or central cavity. This is one of the important point or important question which is getting asked usually. Okay, these vessels, as we have discussed, these are multicellular component. In this multicellular component are made up of made up of many xylem vessel element. Okay, and these are multicellular as well as dead. So, and this particular particular are connected with each other somewhat. So we can say this is a dead syncyte. Here syncyte is a structure. Syncyte is a structure formed by fusion of cells. Then it is known as syncyte. And it is obviously a dead cell, so we can call it as a dead syncyte. Okay. Now, if we go on talking further, we come to know that the end walls, suppose this is we can say one xylem vessel elements, then another xylem vessel element is present like this. So this is the common wall between these two. And in this common wall, if you see, you will see the pores. You will see the pores. Okay. These are the end walls. Okay. And these two common walls, and here also you will see the presence of pores. So here pores are present. So here end walls we can say are perforated. And here, due to the wide lumen, we can say the vessels are more capable of conduction than the tracheids. And another thing, the perforated end walls are there, so pores are completely free to pass the water and mineral, so they work as a pipeline for the conduction of water. So their end walls are perforated, and through this perforation, this perforation, they are connected. Okay. Now perforation may be simple or multiple, means simple pore, one pore B can be there, there can be a many pores. And the contain vessels contain simple pits on their lateral walls. So these are the things which are seen in case of the particularly vessel members. You can see this vessel, this is one vessel element, this is another vessel element, and they are connected through walls, end walls. This is the thick lignified cell wall, and apart from this, they have what, a wide lumen. So this characteristic help to identify us what are vessels or what are trachea. Now after this, if you go on talking about this in detail, then we talk about this, what occurrence. We talk about occurrence. And when we talk about this occurrence or presence, we call that vessels, presence of vessel. Presence of vessel is a characteristic feature of angiosperm. And usually vessels are absent in gymnosperm, but exceptionally present in some gymnosperm like ephedra gnetum and velvischia. These three are the members of order Gnatels, last advanced order of gymnosperm, as we have discussed. Yes, vessel is also a characteristic feature of angiosperm, but we can't say that vessels are found in all angiosperms because vessels are absent in some of the angiospermic families are absent in some angiospermic families like Winteraceae, Trochodendroceae, and Tetracentraceae. These are the three families in which you will not see the presence of these vessels. They are also absent in some plant-like and plant-like Dracena and Yucca. In this also they are absent. So this is all about the what vessels, and the trachea and vessel which we have discussed till now. They are the tracheiary elements of the xylem, and in angiosperm, these two are the main water transporting elements. That are the vessels and the trachea. After this, now we are going to talk about the xylem fiber and the xylem parenchyma. But for now, we will discuss first xylem fiber. So next we are talking about xylem fiber. This xylem fiber have thickened walls, they have thick lignified walls, and due to the highly thickened cell wall, they have a almost reduced lumen, and for the almost reduced central lumen, the term which is used, that is obliterated. So the obliterated means almost reduced central lumen is found. Now when we see the fiber, this fiber may be septate or aseptate. Means so they are either septate or aseptate. Okay, we can say, because fibers are usually found elongated. Okay. Now one more thing which we need to note down here, that this xylem fiber are also known as bast fiber. Oh, sorry, not bast, wood fiber actually. Bast fiber is the phloem fiber. Here they are known as wood fiber, because we will see that wood is a term for the xylem. So they are known as wood fiber. Okay. They may have a cross wall or may cross wall because they are found somewhat like this elongated. So they have a septa or cross wall in this, in spite of presence of a thick walls. Then they are known as septate, but such cross wall are not found. Then they are a aseptate fiber are seen. Okay. Now, if we go on talking about this xylem fiber further, then we come to know that they have highly thickened cell wall and obliterated central lumen, and they are elongated with tapering ends, elongated with the tapering ends they are found. Apart from this, you will see that they provide, if you talk about the function, they provide strength to the tracheids and vessels. And vessels. And if we talk about that occurrence of this xylem fiber, then they are abundantly found in secondary xylem. Abundantly, or in many number, they are found in secondary xylem. And secondary xylem itself, it is known as wood. One more thing is that they are generally not found in gymnosperm wood, because gymnosperm wood is a soft wood. So they like this particular gymnosperm wood, this particular xylem fiber. Okay. Now after xylem fiber, the next element of the this xylem is going to discuss, that is xylem parenchyma. And as we have discussed, this is the only living member of the xylem. So the cells of this xylem parenchyma are living as well as thin walled. They are living as well as thin walled, and the cell walls, thin cell walls, are made up of cellulose. Their cell wall are made up of cellulose. One more thing which is seen with respect to function, that these are the living cells. They have a thin wall, they have a cellulosic cell, and the function which they carry out, that is the storage of food material in the form of starch or fat. And they also store some of the other substances like tannins. Apart from this, if we talk about xylem parenchyma, then xylem parenchyma is also known as also known as ray parenchymatous cells. Now, why they are known as ray parenchymatous cells? Because because they carry out radial conduction of water. And due to radial conduction of water, they are known as ray parenchymatous cells. Okay, or sometimes these ray parenchymatous cells are also known as xylem rays. Okay, now after this xylem, all four components which we have discussed. Now, just we have discussed the ray parenchymatous cells. Now, primary xylem. So this is all about the component of the xylem. Now we are going to talk about the types of primary xylem. The types of primary xylem, if we talk, the types of primary xylem are of two types. And when we talk about this two type of primary xylem, they are protoxylem and the metaxylem. The first formed primary xylem is known as protoxylem, and the later formed or secondarily formed primary xylem is known as metaxylem. So whatever the primary xylem which is formed first, it is protoxylem. The laterally formed, it is known as metaxylem. Now, in stem, if you see, you will see that there is a arrangement of protoxylem and metaxylem. And in this, protoxylem lies toward the center and metaxylem lies toward the periphery. So when protoxylem is present towards the center or the pith of the particular stem, then it is known as endark arrangement. Then it is known as endark arrangement. And this endark arrangement is seen in case of the stem. And you can see here in this particular part. See, this is the large cavity containing body are a metaxylem, and this is the small cavity containing protoxylem. So when this protoxylem are towards the center and metaxylem are towards the periphery, okay, then they are known as what? A particularly endar, which is found in stem. Be remember that protoxylem has a narrow cavity or narrow lumen, and periphery of the organ metaxylem is there. This metaxylem, if we talk, that metaxylem have are large sized and they have wide cavity. Okay. Now, apart from this, in roots, if you see, in this protoxylem lies towards the periphery, not towards the center. In initial stem, it is found towards the center, but in root, it is found towards the periphery. And metaxylem lies towards the center. Then it is known as exarch arrangement. And this exarch arrangement you can see here or locate you can here that this is the metaxylem, which is a large size, and this is a protoxylem, which is a small sized. Okay, so small size are present towards the periphery, then it is known as protoxylem toward the periphery, and it is exarch arrangement, which is seen in case of the root. Now, after this, the second one is the phloem, and when we talk about the phloem after a xylem, it's another conducting element, but this is the conducting element for the food. And this phloem transport food material usually from leaves to the another part of the plant. Why? Because in leaves the synthesis is going to occur. So food gets synthesized in the leaves and then it is transported to the other parts of the plant. So it is known that phloem performs the multi-directional transport. Now phloem is angiosperm consists of the four elements or four type of cells because it is of a complex permanent tissue, so it is going to be made up of different type of cells. Now in this, it is made up of four type of cells, that is a sieve tube element, then companion cell, phloem parenchyma, and phloem fibers. Out of this, you will see that this three, that is sieve tube element, companion cell, and phloem parenchyma are the living component, whereas phloem fiber is a non-living or dead component. Okay. Now we are going to discuss this all, but the conducting part is for a conducting part of this phloem, the term which is used, that is uh, lepton. Now, for the conducting parts of phloem, the term lepton is used. Lepton term is used for the conducting parts. So lepton represents what? Conducting element of phloem. You should remember that term phloem is coined by Nageli. Okay. Now after this phloem, if we talk this four component or four element which we have discussed, or four components that are arranged or associated like this in the phloem, and you can see here this elongated part, this one is this all represent what? Sieve tube. And sieve tube is composed of so many elements. These elements are arranged one above the other. Then there is a presence of what? Companion cell which are associated nearby the sieve tube element. Then they have a phloem parenchyma, and then they have phloem fiber arrangement like this. So these four are arranged close to each other nearby. First in this we are going to discuss the sieve tube. Now we remember sieve tube is a multicellular structure, but it is made up of several sieve tube elements. So sieve tube elements are the unit of this particular sieve tube. In gymnosperm, you will see that there is an absence of sieve tube, and in place of sieve tube, we will find a presence of sieve cells in the gymnosperm. But in angiosperm, you will see the presence of sieve tube. Same way, albuminous cells are present in the gymnosperm, but in angiosperm, you will see the presence of companion cell, and that we will discuss later on. So they lack sieve tube and the companion cell, but albuminous cell and the sieve cells are present. Sieve tube element, if we talk, they are long tube-like structure and arranged longitudinally. This sieve tube element is unicellular structure, but they are arranged one above the other in a longitudinal fashion, and they are forming a multicellular structure. They are associated with the companion cell nearby. Further, if we go on talking about some of the points, their end walls are perforated in a sieve-like manner. There is a sieve-like manner in that the end walls. And this end walls which are perforated are known as sieve plates. The function of sieve tube are controlled by the nucleus of companion cell. Actually, the nucleus is absent in the sieve tube element. So the functions are controlled by the nucleus present into the nearby companion cells. If we talk about the mature sieve tube, they possess a peripheral cytoplasm, but they have a large vacuole, and due to the large vacuole development, the cytoplasm become peripheral at a periphery, but nucleus get absent at the maturity, so they are going to become a particularly a structure that lost nucleus at the end. So these are the things. So we can suppose this is a sieve plate. This is the sieve tube element. This is again a sieve plate, and this is again a sieve tube element. So such sieve tube element are going to form sieve tube, and here you will see the presence of what? Particularly this at end walls sieve plate. Here also you will see the presence of sieve plate. So if we talk about a characteristic of sieve plate or a sieve tube, this sieve tube elements are joined by end walls and that are perforated. They have pores. They are sieve plates. If we talk about sieve plates, they are present obliquely. means they are present somewhat obliquely. And mainly the translocation of fluid material takes place through this pores. Now this sieve tube, if you talk, it is a living inside. It is a living inside, means it is a living cell, and the multiple cells are fused and forming this sieve tube. The walls are thin. The sieve tube have a thin wall. Actually, exactly we can call it as a sieve tube element characteristic. They have anucleated, they are without nucleus, and all other functions we have already discussed. But one of the points, some of the points which are important to note down with this, that is about the callose deposition. When we talk about callose deposition in case of the sieve tube elements, this take place on radius of sieve pores. On radius of sieve pores during dropping season, during dropping or falling season of leaves. And that is autumn. That is autumn. And this deposition in the sieve pores known as or called as callose pad. Okay. Now this callose pad actually protect sieve plate from bacterial infection and dryness. This way, this callose pad protect this particular sieve plate. Actually, this callose dissolve during the spring season. Actually, this callose dissolve during spring season, and this callose is actually a polymer of beta 1,3 glucan, means glucose are going to be linked by beta 1,3 linkage. Apart from this, they have a special type of P protein. P protein, here P means phloem. Phloem protein are there. These are the special type of protein found in the sieve element. And the function, if we talk, the function is the sealing mechanism on wounding along with callose, and it also related with the conduction of food. It also help in conduction of food. Now, in pteridophyte and gymnosperm, sieve cells are present. So, in pteridophyte and gymnosperm, sieve tube is absent, but the sieve cells which are going to be present, that sieve cells are arranged in zigzag manner. They are arranged in zigzag manner. Okay, so this is the thing which is seen in case of the sieve tube. Now, after this, next we are going to discuss the another component of the phloem, that is companion cell, which are present side by the sieve tube element. But these are parenchyma, specialized parenchyma cells, and they are found closely associated with the sieve tube element. The sieve tube element and the companion cell are connected by pits on their common longitudinal wall. The common wall which is present between the companion cell and the sieve tube element, you will see the presence of some unthickened areas, and that are the pits. And actually, through these pit fields, the nucleus of companion cell maintains the pressure gradient or function of the sieve tube element. So the companion cell help to maintain the pressure gradient, or whatever the flow of food is occurring, during that to maintain the pressure. It is important role, and that is maintained by the nucleus of companion cell. Now, apart from this, if we talk, it is known that they have an elongated large nucleus, and the function also we have discussed. Now, it is known that one more important point with this, that companion cell and sieve tube element companion cell and sieve tube element are known as sister cells because both are ontogenetically related, means originated from single mother cell. Okay, and they are known as sister cells. Now, the companion cell found only in case of the angiosperm, they are absent in gymnosperm and angiosperm, gymnosperm and pteridophyte. So, in pteridophyte and gymnosperm, in place of companion cell, albuminous cell or Strasburger cell are present. Now, after discussion of this companion cell, now let's talk about the phloem fiber. So, okay, before phloem fiber, we are going to discuss phloem parenchyma. Sorry. So, phloem parenchyma is the third component, which is a living component again. And this living component is also made up of elongated, tapering, cylindrical cells with a dense cytoplasm, and they have a nucleus. So, they are living cells. They have a thin wall of this. And you can see here that according to this, the phloem parenchyma is here somewhat. Here also you can see this enlarged view. They have a dense cytoplasm and the nucleus. So the cell wall are composed of cellulose, obviously. And apart from this, they have pits through which the plasmodesmatal connection exists within the cells. They all are connected with each other through the plasmodesmata. Apart from this, the phloem parenchyma, if we talk about its function, it stores food material. And the main function of this phloem parenchyma is to store the food. In food, apart from food materials, other substances like resin, latex, and mucilage are also getting stored. Phloem parenchyma is found to be absent in most of the monocot. B represents reporter statement. They are absent in most of the monocot. This phloem parenchyma, from which you will see the stored material are releasing like this, and they can be collected. This parenchyma, also known as ray parenchyma, also known as the ray parenchyma, as they carry out radial conduction of food, as they carry out radial conduction of food, means sidewise conduction of food. Okay, so this is the things with respect to the phloem parenchyma. Now we are going to talk about the last component of this phloem, that is a phloem fiber. And this phloem fiber are the dead cells, which are also known as bast fiber. And obviously, if they are a dead cell, they are made up of sclerenchymatous cells. They are generally absent in the primary phloem. They are absent in primary phloem, but they are found in the secondary phloem mainly. And they are much elongated. They are unbranched, important point. They are unbranched and they have a pointed and needle-like apexes or apices. And you can see this is the phloem fiber with the needle-like or pointed apices. Okay, and they are made up of sclerenchymatous cells. They are unbranched. And the wall are thick due to the lignification and due to which they are a dead cell. They are a dead cells. Okay. Now, if we talk about them in further detail, the cell wall of phloem fiber are quite thick. At maturity, you will see that these fibers lost their protoplasm and become dead. If we talk about the phloem fibers, which are of jute, flax, and hemp, are used commercially. Commercially in market, whatever the jute, flax, and hemp we get, they are nothing but we are getting the phloem fibers. Okay, which is the important thing. And the first formed primary phloem consists of narrow sieve tube. First from primary phloem, which have a narrow sieve tube, they are known as protoploem. The later formed primary phloem have a bigger sieve tube and they are known as metaploem in case of the primary phloem. Okay, so this is all about what a particularly a phloem fiber and the particularly for protoploem and metaploem, which are parts of primary phloem based on origin. First from primary phloem is protoploem, secondary later from the primary phloem is metaploem. Later on, flex fibers you will see, these are the example of phloem fibers. Hemp fiber, you whatever see, that are also phloem fibers. Okay, so I hope you understood this phloem in detail. Let's discuss some of the questions. Xylem and phloem are. They are obviously a complex permanent tissue. Then in flowering plants, tracheids and vessels are. In flowering plant, tracheids and vessels, if we talk, they carry out conduction of water and minerals. And they are main conducting, transporting elements for the water and mineral. When we talk about a long cylindrical tube-like structure in xylem, which is made up of many cells, then it is obviously a multicellular structure, that is a vessel. Actually, vessel member is a single one, which is made up of many cells. Then it should be the vessel should be the answer. Vessel members are going to form vessel. So vessel should be the answer. 3 is not answer. First from primary xylem elements are called protoxylem. Which tissue transport food material from leaves to other parts of the plant? Then obviously it is going to be the phloem, which carry out multi-directional transport. Okay, so this is all about the complex permanent tissue, that is xylem and phloem. I hope we have discussed all things, that is its particular different component, their structure, their function, in case of dead element, in case of living element, all this we have discussed. So I hope you understood well. Thank you. Thanks a lot.